Well, thank you all for being here for this workshop and for sticking around for my workshop overview. Again, I'll be talking about plant de novo assembly with nanopore reads, but I'll also be talking about some of the challenges that plant biologists face when trying to sequence and assemble plant genomes. So when we first consider generating an assembly for a plant species, as you may well imagine, there are a number of special considerations to make, particularly if there isn't already an existing high-quality reference. So for instance, your genome size could be quite small, from something like the 61 megabase pair Genlycia tuberosa, which is a little bladderwort native to Brazil, to something like a 160 gigabase pair genome of the small New Caledonian fern that was recently in the news last summer. It's also critically important to know the ploidy of the plant. So this is, as you saw with Kevin's work, can be a really key challenge to phasing genomes. And many plant species have higher ploidy numbers or even mixed ploidy, such as the commercial strawberry, which is an octoploid. In addition to polyploidy, we have things like repetitive and transposable elements, which are quite ubiquitous in plant species. Things like the maize genome, as I'm sure you're familiar, can be up to 85% repetitive elements. And these pose really unique challenges to assembling genomes. Likewise, these transposable elements can, be typically on average about 10 kb. This is fine if your sequencing technology can handle, up to 20 kb. But there are things like LTR retrotransposons that are very common in plant species that can be much larger than this, so up to 30 kb. And in order to characterize TEs of that size, you really need a sequencing technology that can handle read lengths like that. High heterozygosity is likewise another one of these genome complexities that can cause challenges to resolving plant genomes. Now, beyond that, things like secondary metabolites truly pose unique challenges to plant species. This is true as well of many animal species. Insects can have issues as well. And this in particular is an issue when we're talking about rgDNA extraction. So often you have things like polysaccharides that like to co-precipitate with your gDNA, and you may want to consider things like your sample quality or even your sample preservation methods in order to try to alleviate some of these secondary metabolites. So things like etiolating your leaves in advance of gDNA isolation can alleviate some of this to some extent. Finally, the last challenge when thinking about not just plant life, but all non-model species is the diversity of samples. So I think most of us who've done any sort of non-model research have wished for an easily optimizable DNA extraction protocol, and that often, unfortunately, is not the case. So I'll be going through some helpful tips for plants to at least move us closer to that kind of optimization that we're after. And so I want to show this table here to really drive home this idea that what you're missing in your sequencing does matter. So as we digest this table that you see, you'll see different genomic features that are listed on the left, and then current technologies listed across the top. To orient you, these check marks are representative of the usefulness of a particular technology to achieve precision in that particular area. And so the darker check marks mean that there's some partial usefulness in those particular areas. So in this example, just to orient you further, we typically refer to large structural variants or complex structural variants as those greater than 20 kb in length, or those that have complex rearrangements. We know that short reads are effective in detecting small variants, and we have several tools on the market that have been developed to detect those. Accurate longer reads of 20 kb or less can add detection of those shorter structural variants and full-length isoforms. But in order to get at these longer structural variants that we see, you really need a long-read technology that can achieve greater than 20 kb lengths. Moving along, we know too that things like chromatin conformation and optical mapping are great at getting at slightly more complex structural variants. And we know too that they can be beneficial in large repeat regions and transposable elements. But where a lot of these technologies fall apart is in the centromeric to telomeric regions. And as we heard from Kevin, being able to span across those regions. And then finally, when we think about epigenetics, many of these technologies, you have to incorporate multiple omics in order to get at methylation, in addition to your canonical sequencing information. And they may not include a variety of contexts of methylation or a variety of methylation types. And so what I'll show you in a moment is that nanopore is capable of doing that. So again, methylation is quite ubiquitous in eukaryotes. We know that 5-MC in particular is quite well characterized as an epigenetic mark, particularly in humans and animal species. We know that that's involved in biological processes such as gene silencing and imprinting. But that particular methylation mark alone is not solely important in plant species. 5-MC in all contexts, so your CG, your CHG, and CHH contexts play very crucial roles in regulating gene expression, genome stability, and adaptation to environmental cues in plant species. And so one of the notes there on the left-hand side of the screen is mentioning maize in particular, and the CpG methylation in transcribed regions is positively correlated with transcription, whereas CHG is negatively correlated. And so with nanopore and our powerful base caller Dorado, we have the capability of ascertaining all context methylation in 5-MC, as well as 5-HMC and 6-MA in DNA sequences. So in addition to performing an assembly on DNA, to generate a complete picture of a genome, it's essential to analyze transcripts in order to be able to annotate it. This analysis may come from different tissues, but the most important aspect of transcript analysis is to be able to detect different isoforms. With nanopore, you can achieve full-length isoform resolution. You can achieve alternative splicing information, isoform switching information, gene fusions, transcripts targeted by nonsense-mediated decay, and you can perform some missing potential discovery, as well as missing pathogen resistance that aren't achievable with other technology types. And so what we have here with nanopore is a new way of defining an unbiased complete genome. I've mentioned epigenetics and full-length isoforms already. I'll move forward talking more about high-quality assemblies, and you can imagine from a single high-quality assembly, you can conceive of assembling pangenomes as well. So let's get into the nitty-gritty of it. This is something I, in my own research, spent a lot of time thinking about. I dealt with many ecological samples, plants that ranged from diploid to octoploid in polyploidy, and thought a lot about optimal extraction methods for those species. What I found, and what we have found at Nanopore, this is probably not surprising at all, is that extracting from fresh or flash frozen tissue is ideal over other methods like silica drying, for instance. This is particularly relevant when we're talking about long read technology, as you want to limit the fragmentation and degradation of your sample as much as conceivably possible. So when we're thinking particularly about obtaining a high molecular weight DNA, we have found that a CTAB-based extraction using Carlson lysis buffer, followed by purification step with QIAGEN genomic tips, really yields great quality DNA. And I can't stress enough in terms of sequencing, not just on a Nanopore platform, but on all platforms, that what is most important is starting with a high quality gDNA sample. Your life is made much easier downstream if you put a lot of consideration into this prep step. So depending on then the quality of your DNA, you may also include an optional size selection. If you have high yield and you have very nice non-degraded sample, size selection can increase your read length in 50. But again, this is optional if you are dealing with a more degraded sample. So moving forward into library prep, I know many people are familiar with our ultra-long sequencing kit, but as Kevin mentioned, and also Sean, what we are finding is that our ligation sequencing kit is really quite useful at getting high quality sequencing data. Nice read in 50s, up to 50 kb, and really not sacrificing accuracy or output using this particular method. So this is a 60-minute PCR-free library prep. I, again, spent my graduate school research doing bisulfite sequencing, which was a very long, protracted method, and was shocked to find that I could do this in 60 minutes with nanopore sequencing and achieve canonical and methylation information. So this, again, is very optimized for high accuracy and output, as well as depth of coverage across plant genomes. It's great for things like limited sampling, challenging genomes, challenging extractions, and higher ploidies. And so skipping sequencing just for a moment to talk about analysis. As has been mentioned, the HiFi ASM now has an ONT option. So once you've generated that sequencing data, we have quite a straightforward series of analysis tools that we recommend be employed to both assemble and polish your genome. So prior to genome assembly, we recommend using SeqKit to filter reads with a quality score of over 10. And then for assembly, as I mentioned, we recommend using the assembly tool HiFi ASM with settings optimized for simplex assembly using Oxford Nanopore reads. Now, if you have degraded samples, we do have an optional assembly method. This is Fly. And this is recommended for read length in 50s below 10 kb. It is quite robust across a variety of species and data sets, though handling of ploidy can be limited in highly heterozygous genomes. In both instances, we recommend using Medaka for polishing, and this can enhance the overall assembly quality. Oops. Now, jumping back to the sequencer itself, many of you are surely familiar with our famous MinION devices. You might be familiar with our workhorse GridION device. But when we're thinking about whole genome sequencing of complex genomes, large genomes like plant genomes, the PromethION family is really where you want to put your thought and energy to generate this kind of sequencing. So we have two PromethION 2 devices, the P2 Solo and P2 Integrated. These devices cater to labs that might not have a really high throughput or high volume of samples that they're sequencing in a given year, up to something like the P24 or P48 if you're doing enormous high volume of sampling, generating whole genome sequencing samples. The other thing I want to highlight is our transparent pricing options. There are options available for the number of flow cells that you'd like to receive. We have packs up to 24, and we have options to receive those flow cells over the course of a 12-month period in order to suit your specific needs. Now, what I'm really talking about here today is that we have kind of a new, not just kind of, we absolutely have a new way forward in sequencing approach. So this nanopore approach is a multi-omics approach using a single platform to get at a wide variety of questions. What you see on the bottom of this slide is the current multi-assay omics approach, where you may have multiple sequencing devices, multiple libraries that you have to construct, and then integrate downstream of that, that really cuts into the amount of time that you're spending in wet lab practices and in bioinformatics processes. And so the solution that we offer is a single prep type, a single sequencing run, and a single analysis workflow on one platform to get at all of these same structural variants, methylation, phasing, anything that you would need to construct a genome and then look at isoform information as well. Now, when we talk about one sequencing sample, one flow cell, what I mean is a 30x coverage sample on something like a 3 GB genome. If you want more coverage, you can imagine that you can add and run multiple flow cells in tandem to achieve the coverage that you desire for the complexity of your genome. And to add to this, one thing that I'm quite proud of that Oxford Nanopore has done, not just in terms of providing a multitude of online resources to put the training in your hands, but over the last year or two, we've put a lot of effort into a commercial support staff. So that includes people in my role, the field application scientist, as well as a wonderful marketing team and technical application scientists, bioinformaticians, et cetera, who are here to help you, guide you through your project. So come find me if you're interested in talking about optimization. If you're interested in talking about your individual species type, we are so willing to put this effort toward really making this the best for you. So in terms of our online resources, we have training videos, we have a wonderful protocol library and a protocol builder that's available on our community page, close to 100 step-by-step protocols. This includes several gDNA protocols for unique species. You saw on the earlier slide that there was a fever tree extraction protocol, but we have several for plant leaf gDNA for you to take a look at. And then we also have end-to-end workflows. So lots of bioinformatics and analysis tools that really make your life much simpler. And I just want to highlight this briefly. Kevin's work, I think, covered this to some extent. This is another T-to-T assembly. This is Sergei Korin's work, which you may have been in his talk earlier. Now, what I spoke of before was not a T-to-T assembly, but you can imagine here that the sky is the limit in terms of your capabilities with our technology. So with LSK and the sample best practices that I spoke about, you can still assemble beautiful high quality genomes, but you can even go beyond that. And so I'll just say here, the highlighted column is the Q value. And so for tomato and maize, they were yielding over Q values of over 50. And you'll look as well on the far right-hand side, or excuse me, far left-hand side, that the contig sizes or contig numbers are quite small. And so I think, again, Kevin got at this in his talk, where you're dealing with very long reads. They're much easier to phase, much easier to resolve. And so I just wanted to leave you with this and allow you to think more about what you can do with our technology. And so you saw this slide already, but I think that this is a great closeout for us. Our technology is capable of achieving, what small read technology can, what 20 kb technology, chromatin conformation, optical mapping can, and you can get even more. And so thank you for your time.